Perhaps you know how normal body cells replicate. One cell splits into two identical replicas of itself through the process of mitosis. But for sex cells, the cells that allow for reproduction, this process is different. Sex cells are the egg for females and sperm for males. They don't replicate using mitosis. Rather, they reproduce themselves using something called meiosis. The key difference between meiosis and mitosis is that meiosis prioritizes genetic variation. Genetic variation isn't really needed in the body cells, since they're all your own as an individual. But sex cells make new organisms entirely. On a species-wide level, higher genetic variation means higher chance of survival for the species. If an organism has a mutation to survive a contagious disease that the others don't, then this organism will be able to survive and keep the species alive in the end. Thus, to keep up the genetic variation, meiosis will produce 4 genetically different cells instead of 2 identical cells. These cells will all have half the amount of DNA that a normal cell will have. In humans, this is 23 chromosomes. We call these haploid cells. Cells with a normal amount of 46 chromosomes are called diploid cells. But first, to understand how we get to the final product, we have to see all steps of meiosis. Meiosis has 2 stages unlike mitosis, and has meiosis 1 and 2. One note about chromosome counts. When counting chromosomes, we do not count them by the number of chromatids or each side of the chromosome. Instead, we count them by the centromere, the structure connecting the two. This means that a chromosome that looks like this will be counted the same as a chromosome that looks like this. A normal body cell or diploid will have 2 N chromosomes, and a haploid cell will have N chromosomes. N is a variable used because every species has a different number of chromosomes in its cells. Meiosis still undergoes interphase, that being G1, when the cell duplicates organelles and grows in size, S, when the DNA is replicated, and G2, its final growth stage. The first step after interphase is prophase 1. DNA replicated in interphase is not in its chromosomal form, so this phase's purpose is to condense the DNA back into chromosomes. In this stage, the singular cell will have 2 N chromosomes. What also occurs in this stage is a process of crossing over. The chromosomes begin to pair up with their homologous chromosomes. Homologous chromosome pairs are 2 chromosomes with the same structure, centromere placement, and that code for the same traits. These chromosomes do not have to be perfectly identical, and in fact, them being unequal is the entire point. Parts of the chromosomes that code for the same traits swap with each other. This means that the result are 2 chromosomes with different overall genetics than before. The purpose of this is to increase genetic diversity and enhance species survival. The second phase is metaphase 1. The homologous pairs will line up in the middle in random orientations and be attached to spindle fibers at the polar ends of the cell. The third phase is anaphase 1. This is when the spindle fibers pull the homologous chromosome pairs apart to the polar ends. In telophase 1 and cytokinesis, the cell splits into 2, each only with the chromosomes at the ends of the cell. A new nuclear envelope forms around them. The resulting cells will be haploids and only have N amount of chromosomes. Keep in mind that both sister chromatids are still here. In meiosis 2, we'll be splitting them apart to form the 4 cells. Next is meiosis 2. This stage generally follows the same processes as mitosis. In prophase 2, the chromosomes recondense again, and the nuclear envelope breaks down if needed. In metaphase 2, the chromosomes line up again. In anaphase 2, instead of the chromosomes being split up this time it's each individual sister chromatid. The chromatids move to the ends of the cell. In telophase 2, the cells split. The end result is 4 total cells, each a haploid. In humans, they will have 22 chromosomes. The key thing to note is that each individual chromatid still counts as one single chromosome, and that's the main reason why the math doesn't add up. Thank you for watching. If you have any feedback, please leave it in the comments below. And thank you everyone for watching.